a major breakthrough in depression research, specific brain cells identifying the cellular basis of depression. In a significant advance for neuroscience and psychiatry, researchers at McGill University and the Douglas Institute have identified specific brain cell types that exhibit altered behavior in individuals with depression. This finding provides one of the most detailed biological insights into the disorder to date and may guide the development of more precise, targeted therapies. If you like my videos, subscribe my channel and press the bell icon to turn notifications on so you keep getting all the major advancements in science and technology. And don't forget to like and share this video with your friends and colleagues. The study, published in Nature Genetics, demonstrates that depression is associated with measurable changes at the cellular and molecular level, reinforcing the understanding of depression as a neurobiological condition rather than solely a psychological one. A cellular-level approach to mental health. Depression, which affects hundreds of millions of people globally, has long been studied through behavioral and clinical observations. However, identifying its precise biological origins has remained challenging due to the complexity of the human brain. To address this, the research team employed advanced single-cell genomic techniques to analyze post-mortem brain tissue. This approach enabled them to examine gene activity at the resolution of individual cells, providing a level of detail that was previously unattainable. The study utilized samples from the Douglas Bell Canada Brain Bank, a rare and valuable resource containing brain tissue from individuals diagnosed with psychiatric conditions. By comparing samples from individuals with and without depression, researchers were able to isolate cellular differences associated with the disorder. Two key cell types implicated. The analysis revealed significant alterations in two distinct populations of brain cells, excitatory neurons, which are critical for regulating mood, cognition, and responses to stress, microglia. Specialized immune cells in the brain, responsible for maintaining neural health and modulating inflammation in both cell types. Numerous genes displayed altered activity patterns in individuals with depression. These changes suggest disruptions in neural signaling and immune-related processes within the brain. Excitatory neurons are essential for transmitting signals across neural networks, and their dysfunction could impair emotional regulation and stress responses. Meanwhile, altered microglial activity points to a potential role of neuroinflammation in the development or progression of depression. Linking gene regulation to brain function. A notable aspect of the study is its integration of gene expression data with epigenetic regulation, mechanisms that control how genes are turned on or off without altering the underlying DNA sequence. By mapping both RNA activity and chromatin accessibility, the researchers were able to identify not only which genes are affected, but also how their regulation is disrupted. This dual-layer analysis provides a more comprehensive view of the molecular processes underlying depression and helps explain how environmental and genetic factors may interact to influence brain function. Implications for understanding depression. The identification of specific cell types involved in depression represents a conceptual shift in how the disorder is understood. Rather than viewing depression as a diffuse or generalized brain dysfunction, the findings suggest that it arises from targeted disruptions within defined cellular systems. This reinforces a growing consensus in neuroscience. Depression is rooted in biological processes that can be measured, studied, and potentially modified. Toward targeted therapies, one of the most promising implications of this research is its potential to inform new treatment strategies. Current antidepressant therapies often act broadly on neurotransmitter S systems and can vary in effectiveness among individuals. By contrast, therapies designed to target specific cell types, such as modulating excitatory neuron activity or regulating microglial function, could offer greater precision and improved outcomes. Additionally, understanding the molecular pathways involved may enable the development of biomarkers for diagnosis or treatment response. Future directions. While the findings mark a major step forward, further research is needed to determine how these cellular changes translate into the symptoms of depression. Future studies will likely focus on investigating how altered cell activity affects neural circuits, exploring causal relationships between gene regulation and depressive behavior, developing interventions that specifically target the identified cell population's conclusion. This study provides compelling evidence that depression is associated with distinct, cell-type-specific changes in the brain. By identifying alterations in both neurons and immune-related microglia, researchers have uncovered key components of the disorder's biological foundation. As this line of research progresses, it holds the potential to transform the diagnosis and treatment of depression, moving from generalized approaches toward precise, biologically informed therapies.